So here's my disclosure. I do work for Caris Life Sciences. I'm not going to talk too much about what we do today, but I'm definitely going to explore some of the things that we're doing with a lot of the data that we generate. So at Caris and broadly, we try to start to think about the world in a different way. The advent of medicine really was symptomatic in the beginning, and we have now evolved our technology to the point where we can begin to understand the root causes of things. And as we do that, it means that we need to change the way that we are trying to identify the underlying complexities of the molecular systems that are driving these adverse events. And so as we start to explore the vast amounts of data that we can now acquire, we need to think about ways of incorporating a multi-parameter, nonlinear association to drive our ability to influence those underlying systems. And so that's going to be at the heart of everything that I am going to try to talk to you about. And it's really, at the end of the day, got to be reduced to an actionable event in the clinic. If we can't inform patient treatment, then it's a research exercise. So at the end of the day, to be clinically useful, we have to be able to impact patient care. So as we start to think about this, what becomes abundantly clear is that what we're dealing with is a multi-attractor landscape, where the complexity of the interacting systems is so great that it's unlikely to be well understood in a linear fashion. And so we need to address this and embrace this. The inherent complexity that exists is not something that we're going to be able to just simply ignore. We have to address it. And so that begins to evolve our thinking about how to invoke as many different responses simultaneously through regimen treatment as possible. And so at the end of the day, when we talk about precision medicine and oncology, that's really what we're trying to do. We're trying to influence those underlying systems in a way to improve outcomes. And that requires a great degree of advancement both on the therapeutic side as well as on the diagnostic side. So what we're trying to do is measure as many different things about patients as we possibly can in order to understand all of the diverse aberrations that are occurring so we can select the appropriate therapies. Right now, we're doing that through a combination of testing at the DNA level, the RNA level, at the protein level. But really, we need to take that and scale it up dramatically. We're still only scratching the surface with the information that is available to us in terms of being able to understand how to modulate these systems. And so at the end of the day, again, it's all about finding the right drugs, the right drug combinations, and invoking those responses in patients at the right time. It's the right drug for the right patient at the right time. So the data set that we are working with today comes from our clinical testing service. It's a 592 gene DNA panel. We're getting single nucleotide variants off of that, frame shift mutations, copy number alterations. We don't look for structural rearrangements there because the deficiency is pretty substantial in terms of differentiating between things like translocations, inversions, deletions, and duplications. For that, we go to RNA. At the RNA level, we just recently launched a whole transcriptome assay. So this is for clinical testing. It's covered by insurance. This is a service that we provide to late-stage cancer patients. And for that whole transcriptome assay, we're able to get all of those structural arrangements in addition to expression profiles and are really able to find far more actionable alterations by going to a very deep sequencing at that level. Then we supplement that with protein level assessments, so looking for things like PD-L1, looking for MMR deficiency, a whole host of different IHCs that are customized on a per lineage basis so that we're maximizing the opportunity for patients to get the right type of therapy. So we can take all this information and aggregate it together and begin to look for patterns. But the key is having longitudinal outcomes data on a large number of patients. We run into a hypersegmentation problem very, very quickly where the uniqueness of each patient profile makes it very difficult to be able to assert whether that's just an artifact or whether that's a true driver of response. And so the data set that we have to work with today is comprised of 160,000 molecular profiles. We've got a biobank of an additional 90,000 patients worth of samples, so we can go back and look at the hypotheses that we're generating and then confirm them with additional tests. And we've got outcomes data on about 20,000 patients. So the outcomes data is on average about 4.5 years old in terms of the maturity. We started collecting that in 2009 and have been tracking those patients ever since. That's going to increase quite dramatically this year. We expect that number to exceed 100,000 patients by the end of this year by going in and mining EMR systems, and I'll show you a little bit about how we're doing that, and the clinical endpoints that are available to us through that type of assessment. But first, I want to talk a little bit about a technology called ADAPT. It stands for Adaptive Dynamic Artificial Polyligand Targeting. I dare anybody to say that fast 5 times. Especially at the end of the night after a couple drinks, it's really kind of humorous. But the whole idea here is that we spend a lot of time focused on areas where we can measure things easily. And so huge advancements in next generation sequencing have allowed us to measure pretty much every single nucleotide in the human genome. And so there's a lot of DNA-based sequencing that's going on. We can measure all 22,000 genes. However, I think what is underappreciated is the inherent increases in complexity as we start to go to the downstream state that is actually regulating the cell. So if you look, there are more than 200,000 unique mRNA transcripts. So there's an order of magnitude increase in complexity as we go from DNA to RNA. RNA is still not driving the underlying system. It's just a stepping stone on the way. When we look at the proteins, there are more than 2 million unique protein isoforms. These things then mix together and converge to form complexes, and we really don't have a good idea how many complexes there are. They're highly dynamic in space and time, and we can come up with back-of-the-napkin calculations like 10 to the 8th to 10 to the 10th to 10 to the 12th, but there is a massive amount of hand-waving going on, and nobody really knows. And what we really need are tools that begin to assess the complexity of that downstream dynamic situation and to be able to look at how that is influenced through various therapeutic interventions. So we're beginning to try to answer those questions with ADAPT. It's not the end of the story, it's just a next step along the way. So the reason why we're doing this is very simple. DNA does not have all the answers that we need. These 2 animals are genetically identical. It is the same animal. In as little as 3 months, a pig that goes and escapes captivity will go through this incredible physical transformation. It's due to an increase in testosterone and stress and dietary changes. And so this is a phenotypic response that we will never find through sequencing. And that's what we need to deal with when we're addressing cancer. It's the end-all, be-all phenotype of the patient's response to therapeutic interventions that needs to be characterized, and only some of that is going to be found at the DNA level. I like to say that we're often looking for our lost keys underneath the lamppost when we're just looking at DNA. And it's been great. It's certainly advanced things, but we need to be thinking about the world in a much bigger way in order to solve the end problems. So the way that we're trying to address these protein aberrant assessments of the system is by using the power of next generation sequencing and using the power of DNA, but in a completely different way. So we like to use aptamers. And the reason why we like to use them is because they create an incredible informatics space that we can play in. And so if you think about building an aptamer, and that's a small single-stranded synthetic piece of DNA that will fold up and act kind of like an antibody, it creates an immense potential. So with 4 bases, if we wanted to make all possible 100-mer random sequences, that would be 4 to the 100th power, or in base 10, that's 10 to the 60th different sequences. So if we were to synthesize just 1 copy of each of those and add up all the mass, that mass is about 10 to the 38th kilograms of DNA. So that's a really, really big number. To put that into context for you, Earth only weighs 10 to the 25th kilograms. So we're talking about 1 copy of each DNA sequence that's 100 bases long weighs more than the entire Earth itself. In fact, that mass is equivalent to about our entire galaxy. It's a little bit smaller than our galaxy, cut off some of the edges there, and no dark matter gets to count in here. But you can just imagine that informatics space. We can start to then apply that to this system of protein complexes. So now all of a sudden numbers like 10 to the 8th and 10 to the 10th don't become so scary because we have an underlying tool that is so vastly more powerful to assess that underlying system. So the way these things work is relatively simple. They've been around for a long time. They've been around for more than 30 years. Larry Gold discovered them back in the early 80s, and they essentially just form an electrostatic hydrophobic knot that can bind to various protein niches. And so the advantages are that, of course, we have a lot of great tools to deal with short pieces of DNA. We can also synthesize them. So they're very easy to make, they're very reproducible. We don't have any of the inherent limitations that exist with the biological systems that we use to make monoclonal antibodies. But they have some deficiencies. There's a reason why they've been around for 30 years and nobody is really using them. And that's quite simple. Their binding affinity is really low. It's about 10 to the minus ninth for KD. We're trying to build individual aptamers that will outcompete individual antibodies, and that's never really going to happen because a monoclonal antibody will have a KD of 10 to the minus 12th. And so if we think about it for a second, it makes perfect sense. Of course, evolutionarily speaking, if aptamers could work better than antibodies, we would have evolved to use aptamers instead of antibodies. There's no loss of complexity. There's no reason why we would be using antibodies instead of aptamers. Energetically, they're much more favorable to create, but they just don't work as well. And so as long as we're trying to do this one-to-one comparison, I think it's crazy. It's just an exercise in insanity. We're doing the same thing over and over again, expecting a different outcome, but it's not going to work. What could work, though, is modeling ourselves after actual biology. In a native system, there's no such thing as a monoclonal antibody. Everything is a polyclonal antibody. That's because nature takes advantage of the opportunity to have multiple ligands bind to the same underlying complex. And so as you attack a complex from multiple sites, then all of a sudden you can deal with the inherent diversity of minor changes in protein conformation that can reduce the underlying KD of any particular binding molecule. And so that's what we can do with aptamers as well. And so instead of having one aptamer trying to bind to a complex, we let thousands or tens of thousands or millions of these things bind to the same underlying system. And as soon as we start to do that, then we can overcome the inherent deficiencies of any one aptamer binding at a low affinity. We use the massive numbers that are available to us in order to overcome some inherent limitations in the kinetics of binding. So let me take you through a couple examples of how we have deployed this technology to date. So the first one is we built an assay to predict drug response as a proof of concept. And the idea behind it is relatively simple. So what we do is we take a vial of randomly generated aptamers, and in this case they're about 90 bases long. And we can pipette up to about 10 to the 12th molecules of these things. So we take our 10 to the 12th unique molecules and we put it on a slide from a patient that has breast cancer that responded well to Herceptin. The sample was selected before they got the drug, so we know what the clinical outcome was. And what ends up happening is a lot of the aptamers bind to features in the cancer, some of the aptamers bind to features in the non-cancer cells, a whole bunch of them don't bind to anything. We can wash everything off and then go in and microdissect out just the cancer cells. In so doing, we're grabbing the subset of aptamers that bound to molecular features present in the cancer cells. We can PCR amplify that and cycle through it. And in just 3 rounds of enrichment, what we see is that we can develop an aptamer library that specifically stains molecular features in the cancer that is not present in the stromal. So what you're looking at here is a DAPI stain, so we can see the nucleus. And then the brown that we add in here is the aptamer library. So at round 0, when we try to light up the aptamers, there are too few copies of any one aptamer that's binding to give us any kind of signal. But after just 3 rounds of enrichment, we now have a library that is specifically staining the cancer of this individual. We can use these as an affinity purification reagent to identify what the targets are. That gives us a whole nother application and source of data. But for right now, we're trying to predict what is the underlying signal that is going to indicate response to Herceptin. So at this point, we don't have that information. We have to take a secondary step. So what we do is we take a second patient. Now this patient does not respond to Herceptin. We take that library that we've now gone through 3 rounds of enrichment on and put that to incubate on the second patient sample. In this case, though, we don't want to keep what binds. We still have lots and lots of elements within that library that are binding to molecular features in the cancer of the non-responding patient. Those are the ones we want to eliminate. So in this case, we keep the supernatant. We keep all of the aptamers that didn't bind to that particular patient's sample. Keep that, put it on another non-responder. You gotta work a little bit harder to deplete these things than you do to enrich them. And then finally, put it back on our responder, dissect out the cancer, and repeat. So after just another 3 rounds of this process, we successfully eliminate the subset of aptamers that are binding to the molecular features that are common between those 2 cancer patients. And so what we have at the end of the day is a library of oligonucleotides, short synthetic sequences that bind to features in the cancer of patients that respond to Herceptin, does not respond or does not bind to the non-cancer cells, and does not bind to the non-responding patient samples. We have no idea what it's binding to at this point, but we don't need to know in order to have a clinically actionable piece of information derived from this. At this point, we can now begin to use this assay to assess, is it broadly applicable? Does it only work on these patients that we trained it on, or does it work across the population? And so we proceeded with the study, and we looked at how effective is this approach in identifying women that respond to Herceptin compared to those that don't. And you can see here is the results of the stain that we created. The dotted blue line here is the control population, those women that were treated without Herceptin, so that's the right control group to make sure this is predictive and not just prognostic. It could just be that we found a stain that tells you you've got a bad cancer. We want to make sure it's predictive. And then the red line here, that's Herceptin. That's HER2. HER2 is only about 45 to 50% accurate in identifying women that will respond to Herceptin. When we look at the clinical outcome, here's the difference in that subset. So these are the test-positive women, these are the test-negative women. And so not only is it something that we can see in terms of staining and diagnostic performance, but it does translate to clinical benefit for the patients that are test-positive. So we published this last year in Nature Communications. It's all there for you to read about in detail when you're having trouble sleeping at night. So the second case is to deploy this technology for a screening assay. And in this case, we're using exosomes as the underlying biological substrate in order to try to identify men that should not go in for a prostate biopsy. So exosomes, for those of you that are not familiar with them, are small lipid-encapsulated bodies that are shed by every single cell type. They are massively abundant. In 1 ml of plasma, there are about 10 to the 10th to 10 to the 12th of these things. So it's opposite problem of circulating tumor cells or of cell-free DNA. The challenge is there are so many of them, how do you find the ones that are telling you about the cancer? Cancer uses exosomes as an intracellular communication method to deploy screening modalities to hide from the immune system, to do metastatic niche preparation. It's really the way that they modulate their microenvironment. So there's a massive amount of information contained within the exosomes if we can find the right subset that can derive it. So in this situation, we basically take plasma from men with prostate cancer, throw in the library, isolate the exosomes. We're then grabbing the subset of aptamers that bind to the exosomes in men with prostate cancer. We take that, we put it in men without prostate cancer, we isolate the exosomes, and we keep what's in the supernatant. And we cycle through that. But at the end of the day, our readout is sequencing based. So what we do is we take the library that we've created, we put it in the plasma of men suspected of having cancer, isolate the exosomes, and then we sequence all of the aptamers. And that's where things get fun from a mathematical perspective, because now all of a sudden we've got between 1 and 10 million unique probing analytes that are telling us about that underlying state for that individual. And we can deploy a lot of machine learning techniques to derive and define what is good about that. And so we've done that. We like to use cross-validation and use permutation testing to make sure we're not diluting ourselves. It's really easy when you're measuring that many things on a small number of patients to find a signal. It's a lot harder to confirm that that signal is real. So the blue lines here are the results of 100 permutations of our cross-validation. The gray lines are when we permute the labels. So permuting is when you mix up, we say this man had cancer and this one didn't have cancer, and then we mislabel them intentionally and see if we can derive a signal. So the difference there tells us that there is a true biological signal being measured that is differentiating men with prostate cancer from men without prostate cancer. We're in the validation phase of this and intend to commercially make this available. It's a decision support tool, so there are about 1.3 million prostate biopsies performed every single year that really only need to be performed on about 300,000 men. So out of the 1.3 million biopsies performed, there are only 300,000 diagnoses of cancer. And then out of that 300,000 men with prostate cancer, only about 30,000 of them will die from the disease. So there's a lot of work that we can do just in terms of the screening modalities to find the right population of patients that need to go in for new prostate cancer therapies, much like what we heard about this morning. And so finding the right patients is critical, and this is one way we can do it. The third opportunity is to find drug targets. So I mentioned we can actually use these as an affinity purification reagent tool to find new novel drug targets. We recently published that, just came out a couple weeks ago. And basically what we did was we found that on the surface of cancer cells, there's a novel spliceosomal factor. And so this is a protein complex, so none of the ways that we have of measuring proteins would work to be able to find this complex on the surface of a cell. And the reason is that when we're doing protein discovery, for the most part, we're using a technology like mass spec. And mass spec is great, and I like mass spec, but it suffers from a fundamental problem. We have to break everything up and chop it up into small pieces, and then measure the small pieces and try and put the problem back together. So when we see nuclear factors and transcription factors, we assume they're coming from the nucleus of the cell. We have no reason to believe that they would actually be on the surface of the cell. And so I like to say, if you were to take mass spec technology and apply it to a Napa Auto Parts store or a Ford dealership, you're going to get the exact same answer, right? You're going to find steering wheels and hubcaps and radiators and transistors, but you have no idea whether you're playing in a parts store or you're looking at a fully formed car. And in the case of cancer and disease, it's more about, is it turned on and is it racing towards the edge of the cliff? Are we looking at a Thelma and Louise type event here? And so being able to assess all of those complexes as they work together is essential for increasing our understanding of what's going on. This particular spliceosomal factor actually is, in fact, druggable. And so the aptamer that we found, if we treat our cells with that, actually induces a necrotic-based cell death by inducing a spliceosomal storm within the cell. So not only is there an opportunity to find novel drug targets, in some cases these molecules may serve as the therapeutic themselves. We don't have high hopes for that. There's an example of this in the past. So Macogen is a drug for macular degeneration that was an aptamer. It got fully FDA approved, and then 6 months later, somebody released an antibody that did much better job of that. So I don't really think that aptamers are the end-all be-all for binding moieties, but they sure do make a great discovery tool. Finally, we can begin to look at all of the vastness and richness of data that we're able to find. So when we did this and we deployed the libraries as affinity purification reagents, we were able to enrich for thousands of underlying protein components that are unique to the cancer, that are not found in the adjacent tissue for those patients. That increases our understanding of what's going on. And so we need to be able to use all of that information that is not currently being exploited, that is coming off the mass spec, to increase our understanding of what's going on. And this is one way of doing that exercise to reduce our signal-to-noise problem in order to facilitate a deeper understanding. So all of this data combined together creates an opportunity for what we like to call next-generation profiling. And so this is application of machine learning to very complex data sets. I am a mathematician by training. I always like to point out that machine learning is not something new from the math world. Most of the algorithms that are deployed are quite old. K-nearest neighbors, the earliest reference to that was from an inventor named Alhazen in the year 1000. So it's not that machine learning is new. What's new is the ability to perform this hundreds of millions of times in a reasonable amount of time. So the computing capacity has enabled us to deploy these algorithms in new, novel ways that allow us to actually deal with super complex data sets. So the way that we're doing this is we're taking our DNA information, we're taking our RNA information, we're taking our protein information, we're defining as many different molecular features as we possibly can, mixing that with our clinical outcome data, and then throwing it into this deliberation engine that we call DEAN. We call it a deliberation engine because we haven't implemented any one of these algorithms. We've implemented all of them. We've got about 400 different versions of each one of them, and then we let them fight. Winner takes all. And so the clinical endpoint that we like to use is time on treatment or time to next treatment. It is not a well-validated endpoint if you go and talk to the FDA, but I believe that it's going to be used more and more in the future because it's robustly captured. If you look at our EMR systems, at our insurance-based healthcare system, the one thing that is really robustly captured is what drugs are given when. And that's because reimbursement is tied to those events. So you know exactly when each patient got what dose of drug. And so if we look at the correlation between either time on treatment or time to next treatment to progression-free survival, we get an R-squared of 0.9. Now, there is some, independent data components to this that we have to be thoughtful of, but we're not asserting causation. We're simply assessing correlation. And so there are risks with that, but for right now, it seems to be working really, really well in terms of predicting patient outcome. The difference between TOT and TNT is dependent upon the type of drug. So we have to watch out for false discovery, definitely avoid that as much as we can, and we do that, again, through permutation testing. So that's where we mix everything up and make sure that we're not diluting ourselves. So when we do these, looking for responders versus non-responders, we see that there's variation in the level of performance of each of our underlying algorithms, but we don't see a similar trend in the randomly perturbed labels. And so at the end of the day, we deployed this in a clinically important setting for frontline colorectal cancer patients in their decision to go on full FOX or full Fury. We built an algorithm, and then we started to test it. So when we started to look at 5-fold cross-validation, we saw some nice signals there. The best models were around 0.31 hazard ratio. A typical model would be more around the 0.47. So we felt like, okay, that's a pretty good signal. Again, we have to check and make sure it's not prognostic, that it's predictive. So this is the cohort of men receiving full FOX. When we look at the cohort of men receiving full Fury, there was no difference. So that confirms to us that this, in fact, is likely to be predictive and not prognostic. Then we have to make sure, okay, is there some right side or left side component that's a big confounder in colon cancer? We saw that the algorithms could define a little difference between right and left, but it was not the primary driver of difference in response. So the signal that was identifying response to full FOX was much bigger than any signal defining right and left sided cancer. So then we ran a blinded study, and we found a hazard ratio of 0.33. So we were able to confirm that, in fact, the signal is real. This is, however, using a non-standard clinical endpoint. So our next step is to run samples from a randomized phase 3 trial to confirm the clinical utility of this signature. Interestingly enough, we saw that there's a similar signature in the adjuvant setting. So this is something that could potentially be moved up in the care paradigm. We've done a similar thing in non-small cell lung cancer for immunotherapy. I think that's been talked about ad nauseum, so I'll just get straight to the point. With that population of patients, there is a subset of patients that are considered hyperprogressors, where the drug actually causes them to die faster. That's a very significant problem. So it's one thing to give a drug that doesn't work. It's another thing to give a drug that causes harm. So we really need to identify that population. So we went through that same process. We identified an underlying signature. We could pick 90 days or 150 days. Either one, we saw a good signal. And then in our validation study, we found that, in fact, it was able to be confirmed, and we can identify those hyperprogressors. One of the most astounding things, though, was in this particular case, we had overall survival as an endpoint as well, and that is a robust endpoint. And when we looked at the patients that had the hyperprogressor signature that were treated with chemotherapy, they were the super responders. So not only is it that the signature is identifying the subset of patients that are likely to be harmed by immunotherapy, that is the very population that has the very best response to chemotherapy. So again, this has to be validated in a randomized phase 3 trial. It's not ready for the clinic, but the intriguing components are there. Finally, we can do this to improve disease accuracy as well. There's enough information to be able to identify what is the underlying source of cancers of unknown primary. And so we can deploy this in a multitude of settings to really enable better patient care. So at the end, I just want to say thank you and hope that you agree that we need a systems biology approach to dealing with oncology. Thank you.
We have time for some questions. I have a question. I was wondering what type of success are you having getting exposome information from the EHR in addition to drug administration?
Very low success.
Okay, that's surprised. And any outcomes other than survival?
Survival and TNT or TOT are the two that seem to be the most robust. Every once in a while, you can get hold of radiological scans and pull out PFS, but they're not time normalized, so it makes it challenging.
Yeah, I have a question about the progression after the immunotherapy, and so there has been a lot of argument and whether that's a real phenomena that really means there are some patients they just don't respond and they're going to progress anyway, and then you see progression. And so what's your view? What's the current the consensus in whether those patients actually are harmed by immunotherapy or they just don't respond to immunotherapy, they progress anyway?
So I don't think that there's consensus. I definitely think it's a hot topic of debate. In the last few months, there have been a couple studies published with similar findings in terms of the hyper-progression phenomenon. There's a robust study that's now being done out of France to truly answer the question. I think that all of the data that exists right now is certainly hypothesis generating and very controversial. It seems likely, just given the traditional response rates to chemotherapy, and you just don't see that many patients dropping off the chart as quickly with chemotherapy. But the nature of it, I think, is not understood at all.
So is that the UCSD study you refer to?
So the first one was out of France, and there was just another one coming out of UCSD with Rizal Kuzrak.
So those are the MDM2 amplified cases you're talking about? And the French group also saw the same thing?
They did,
The MDM2 group, okay.
Yep. Okay, thank you very much.